Hello everyone, this is Mohan Kumar signing in to talk about concept of neurosecretion with few examples. So our main objective includes to understand and learn the basics of neuroendocrine secretion and neuroendocrinology as such, to understand the ways of hormonal expression through an axis. So at the end of the class, you should have a thorough knowledge about the ways in which neurons and endocrine cells integrate themselves to regulate hormone secretion. So in the content section, we have included neuroendocrine cells and their secretions, neuroendocrine integration, and hypothalamus, since it is important organ which we have not discussed elsewhere. So I am incorporating hypothalamus in this section, and the three axes: HP axis, HGP axis, and HPT axis. The neuroendocrine system is made up of special cells called neuroendocrine cells. They are spread throughout the body, and neuroendocrine cells are like nerve cells, like neurons, but they also make hormones like cells of endocrine system, like endocrine cells itself. They receive messages from the nervous system and respond by making and releasing hormones. These hormones control many body functions. So neuroendocrine cells are found in almost all organs of the body. They are mainly found scattered in the gastrointestinal tract, which includes small intestine, rectum, stomach, colon, esophagus and appendix, gallbladder and pancreas, and thyroid gland. Neuroendocrine cells are also commonly found in lungs or airways into the lungs, as well as the respiratory tract of the head and neck. Neuroendocrine cells scattered throughout these organs are often referred to as the diffuse neuroendocrine system. Diffuse neuroendocrine system. The pituitary gland, parathyroid glands, and the inner layer of the adrenal gland are almost all made up of neuroendocrine cells. Other sites of neuroendocrine cells include thymus, kidney, liver, prostate, skin, cervix, ovaries, and testicles. Endocrine cells make and release hormones. We all know that, and similar substances like peptides in response to neurological or chemical signals. The hormones attach to specific receptors on target cells, which bring about changes in the cell and what they do. Neuroendocrine cells have many functions, which include controlling the release of digestive enzymes to break down food, and controlling how fast food moves through the GI tract, controlling air and blood flow through the lungs, controlling the blood pressure and heart rate, controlling the amount of sugar in the blood, controlling the bone and muscle growth and development, so on and so forth. Includes many other functions. And I have a few examples of hormones or peptides released by neuroendocrine cells and what they do. Serotonin. It's also called as 5-hydroxytryptamine or 5-HT. It's a chemical released by nerve cells that helps with digestion. A lot of the body serotonin is found and made in the neuroendocrine cells of the GI tract, where it controls the movement of food through the GI tract. Another example, gastrin. Gastrin tells the stomach to release acid and enzymes to help with digestion. One more example, insulin. Insulin is well known. Insulin is made by the pancreatic islet cells. It lowers the level of sugar that is glucose in the blood when it is high. It controls when cells absorb or take up sugar for energy. Epinephrine or adrenaline is made by neuroendocrine cells of the adrenal gland. It is released during times of stress, like when you feel fear, and increases heart rate and blood pressure, preparing the organism for flight or fight mechanism. Last example, growth hormone is made by the pituitary gland. It promotes growth and development of bones and muscles. The neuroendocrine cells are cells that receive neuronal input, neurotransmitters released by nerve cells or neurosecretion cells like neuronal input. And as a consequence of this input, release message molecules. Here the message molecules are hormones into the blood. This way they bring about an integration between the nervous system and an endocrine system. And the process is known as neuroendocrine integration. The major center of neuroendocrine integration in the body is found in the hypothalamus. As we see, the major neuroendocrine systems here all beginning with hypothalamus. Hypothalamic pituitary adrenal axis, hypothalamic pituitary thyroid axis, hypothalamic pituitary gonadal axis, hypothalamic neurohypophyseal system. Here, hypothalamic neurosecretory cells release factors to the blood. Some of these factors, like releasing hormones, released at the hypothalamic median eminence, control the secretion of pituitary hormones, while others are released directly into the blood. The neuroendocrine system is the mechanism by which the hypothalamus maintains homeostasis, regulating reproduction, metabolism, eating and drinking behavior, energy utilization, osmolarity and blood pressure and so on and so forth. So, since all the axis starts with hypothalamus, and we have studied other glands in our previous lectures like pituitary, adrenal gland, thyroid gland, gonadal gland, adrenal gland, all that we have studied. So I shall focus my next slide on hypothalamus. If you look at the structure of hypothalamus and the labels of it, you will find many nuclei structures like infundibular nucleus, ventromedial nucleus, posterior nucleus, dorsomedial nucleus, paraventricular nucleus, anterior nucleus, supraoptic nucleus, and so on and so forth. So in neuroanatomy, a nucleus, a plural form of which is nuclei, is a cluster of neurons in the central nervous system located deep within the cerebral hemisphere, the brainstem. So this hypothalamus is a portion of the brain that contains a number of small nuclei with a variety of functions. One of the most important functions of the hypothalamus is to link the nervous system to the endocrine system via a pituitary gland. The hypothalamus is located below the thalamus and is part of the limbic system. The hypothalamus is also responsible for regulation of certain metabolic processes and other activities of the autonomic nervous system. So it synthesizes and secretes certain neurohormones called releasing hormones or hypothalamic hormones, and these in turn stimulate or inhibit secretion of hormones from the pituitary gland. So that is why this slide, the axis, the major neuroendocrine system, all begin with hypothalamus and pituitary. So, in hypothalamus, like oxytocin and vasopressin, if we talk about their vasopressin, also called as anti-diuretic hormone. These two neurohypophyseal hormones of the posterior pituitary gland are secreted from the nerve endings of magnocellular neurosecretory cells into the systemic circulation, into the bloodstream. The cell bodies of the oxytocin and vasopressin neurons are in the paraventricular nuclei and supraoptic nuclei. This was also discussed previously. Paraventricular nuclei and supraoptic nuclei, they aid in the release of oxytocin and vasopressin. And the electrical activity of these neurons is regulated by afferent synaptic input from other brain regions. So by contrast, the hormones of the anterior pituitary gland are secreted from endocrine cells that in mammals are not directly innervated. Yet the secretion of these hormones like adrenocorticotropic hormone, luteinizing hormone, follicle stimulating hormone, thyroid stimulating hormone, prolactin, growth hormone, all these hormones remains under the control of hypothalamus. So we discussed about oxytocin and vasopressin under the control of hypothalamus, and now we are saying adrenocorticotropic hormone, luteinizing hormone, follicle stimulating hormone, thyroid stimulating hormone, prolactin and growth hormone. All these are under the control of hypothalamus. The hypothalamus controls the anterior pituitary gland via releasing factors and release inhibiting factors. These are blood-borne substances released by hypothalamic neurons into blood vessels at the base of the brain, specifically at the median eminence. These vessels, the hypothalamo-hypophyseal, not there in the diagram, portal vessels carry the hypothalamic factors to the anterior pituitary, where they bind to the specific receptors on the surface of the hormone producing cells. So the link between the hypothalamus and the pituitary gland is basically through a vessel known as hypothalamo-hypophyseal portal vessel. So an example here, if I have to talk about, the secretion of growth hormone is controlled by two neuroendocrine systems. The growth hormone releasing hormone, GHRH, growth hormone releasing hormone. These neurons, GHRH neurons and the somatostatin neurons, which stimulate and inhibit GH secretion respectively. The GHRH neurons are located in the arcuate nuclei of the hypothalamus, whereas the somatostatin cells involved in growth and hormone regulation are the periventricular nuclei. These two neuronal systems project axons to the median eminence, where they release their peptides into portal blood vessels for transport to the anterior pituitary. Growth hormone is secreted in pulses, which arise from the alternating episodes of GHR release and somatostatin release, which may reflect neuronal interactions between the GHRH and somatostatin cells and negative feedback from growth hormones. So pulsatile release and episodic release of GHRH and the somatostatin would actually aid in the development and growth. The neuroendocrine systems control reproduction in all its aspects from bonding to sexual behavior. This also controls spermatogenesis and ovarian cycle, parturition, lactation and maternal behavior. They also control the body's response to stress and infection. They regulate the body's metabolism, influencing eating and drinking behavior, and influence how energy intake is utilized, that is, how fat is metabolized or mobilized. They influence and regulate mood, body fluid and electrolyte homeostasis and blood pressure. So hypothalamus play a huge role in many of the body's functions. Now, going back a slide, as I said, major neuroendocrine system has these three axes: hypothalamic pituitary adrenal axis, hypothalamic pituitary thyroid axis, hypothalamic pituitary gonadal axis. HPA, HPT, HPG axis. So what is HPA axis? Activation of the HPA axis, which regulates various metabolic functions, is initiated with the release of CRH from the hypothalamus. We see here hypothalamus releasing a CRH. CRH is corticotropin releasing hormone. This release occurs in response to various stimuli, including almost any type of physical or psychological stress during the normal sleep-wake cycle, in response to certain neurotransmitters also. CRH then stimulates the anterior pituitary to produce ACTH, adrenocorticotropic hormone, anterior pituitary producing ACTH. ACTH in turn activates adrenal hormone production, primarily cortisol. So ACTH affecting the adrenal cortex to release cortisol. So cortisol, it can negatively impact the HPA axis by inhibiting anterior pituitary to produce ACTH, or it can also affect hypothalamus to stop synthesizing CRH. Both are possible. So the activity of HPA axis is regulated by negative feedback mechanism, and thus the increased cortisol, as I said, levels repress CRH release by the hypothalamus and ACTH release by the pituitary. So in addition, ACTH can directly inhibit hypothalamic CRH release as well. So there are two checkpoints, as we can see. Cortisol can put one checkpoint at anterior pituitary and another checkpoint at hypothalamus. So master pituitary, master endocrine gland can also be stopped, or the one which regulates the pituitary gland, the hypothalamus, can also be stopped negatively. So this is in HPA axis. What are we saying HPA axis? There is a way in which a target cell reaches the target organ to synthesize a particular hormone and release into the bloodstream, thereby bringing about a certain effect, and it is totally negatively regulated. The next is HPG axis. So in both men and women, the HPG axis, hypothalamus, pituitary and gonadal. So HPG axis. So the HPG axis is the hormone system that controls the release of sex hormones. In both genders, the system is activated by GnRH, which is released regularly in short bursts from the hypothalamus. GnRH then stimulates the release of FSH and LH from the pituitary, specifically anterior lobe of pituitary. In men, LH stimulates certain cells in the testes, like Leydig cells, to release testosterone. FSH and testosterone are key regulators of another set of testicular cells, that is Sertoli cells. Sertoli cells support and nourish the sperm cells during maturation. So LH stimulates testosterone production, whereas FSH stimulates sperm nourishment and maturation. So that is in case of males, whereas in case of women, in case of females, during the menstrual cycle, LH and FSH stimulate the ovarian follicle that contains the maturing egg to produce estradiol. So LH and FSH would stimulate the ovarian follicle that contains the maturing egg to produce estradiol. After ovulation has occurred, LH also promotes production of progesterone and estradiol by corpus luteum. So in this picture, if you look at it, hypothalamus, pituitary and the gonads. So GnRH is released by the hypothalamus into the pituitary, and the pituitary in turn releases two hormones after the reception of GnRH from the hypothalamus, and those two hormones are luteinizing hormone and follicular stimulating hormone. Luteinizing hormone and FSH, both when they reach testes, and they bring about differential effect. In testes it is produced in testosterone, whereas in case of ovary it produces estrogen and progesterone. The last axis is the simplest axis, that is the HPT axis, and the HPT axis is basically hypothalamus, pituitary and thyroid axis. The hormones that make up the HPT axis control the metabolic process of all cells in the body and are therefore crucial for the organisms to function normally. So they increase or decrease the basal metabolic rate of an organism through the production of the thyroid hormone. The secretion of TRH, thyrotropin releasing hormone, from the hypothalamus activates the HPT axis. After reaching the pituitary, TRH, that is thyrotropin releasing hormone, stimulates the release of TSH. TSH is thyroid stimulating hormone, which in turn promotes the production of thyroid hormone at the thyroid gland. Thyroid gland is a butterfly shaped gland located in the anterior part of the neck. So thyroid gland would synthesize two different types of hormone, in fact three, T4 and T3, based on the number of iodine present in the hormone. That is why in the picture, in the diagram, we have represented serum iodine. So TSH would actually induct the iodine into the thyroid as well, activate the follicular cells to release and uptake iodine and release thyroglobulins and manufacture T3 and T4. So TSH plays a huge role here. So thyroid, after receiving the TSH signal, would synthesize T3, T4 and reverse T3 is also possible. T3 and T4 or RT3 is basically labeled based on the number of iodine molecules. T4 has 4 iodine, T3 has 3 iodine molecules. And their production, T4 and T3, are negatively regulated. So the arrow mark goes back to hypothalamus and pituitary as well. So indicating that the T4 and T3 can actually bring in a negative or negate the effect of TSH production or TRH production at either hypothalamus or at pituitary level. So overall, if you look at, the flow is always from the hypothalamus to thyroid, hypothalamus, pituitary and thyroid. That is why it is labeled under HPT axis, hypothalamus, pituitary, thyroid axis. So this is the way hypothalamus is regulating the release of several hormones into the bloodstream and bringing about various actions in the body like hypothalamus, anterior pituitary adrenal. I have just listed out three axes which are most important here. Okay, there are also hormones which I said earlier like oxytocin and vasopressin, which is directly released into the system. So that is part of the hypothalamic neurohypophyseal system, vasopressin and oxytocin. With this, I conclude this session. Thank you very much for your patience hearing. If you have any doubts or clarification, you can leave a message in the comment box or you can reach me on my WhatsApp number. Thank you very much.